First of all, thank you, Keith, Comito, and Lifespan.io organizations for organizing this event. I want to briefly mention that the previous speaker, Peter, is actually a brilliant mathematician, and there are a couple of brilliant mathematicians here. One thing I really like about methylation is that it allows us to bring the forces of math into biology. And today I talk about my favorite topic, epigenetic clocks. Before I do that, I want to address a challenge that several speakers mentioned yesterday. We need high quality data for machine learning, artificial intelligence. And we need full access to these data. And so I'm very happy to announce that we just released data from our mammalian methylation consortium. These are 15,000 tissue samples, 70 different tissue types, 348 mammalian species, high quality data. I attest to it because I was the data manager. If something is wrong, it was my fault. So the data are fully available. They're software scripts. I really invite you to delve into those data. To assemble these data, we needed to assemble a consortium of 200 collaborators. You will recognize some familiar names. The Vadim Gladyshev lab had 10 co-authors, Vera Gorbunova, Juan Carlos Belmonte, many labs collaborated here. We collected data from 25 out of 26 taxonomic orders. It turns out every mammal can be in one of these 26 taxonomic orders. And the one order we missed is the so-called marsupial mole. And I hope one of the audience members lives in Australia. And on your way home, maybe you can look out for roadkill and send it to me. So there's this marsupial mole that we are missing. So talking about the power of mathematics. A mathematician needs axioms, and there will be many debates about what is a biomarker of aging. And here I say these are my axioms, my personal axioms, because people have different axioms. There's no agreement, but axiom number one is it's a quantitative metric that is highly correlated with age. Why? It's called a biomarker of aging. It better correlate with age. Of course, it must be connected to mortality risk and morbidity risk, but in general, it will be different from that. And the reason is a lot of things can affect your mortality almost instantaneously, but they don't age you. The third axiom is it must pertain to a fundamental property or characteristic of cells. Why? Because most cells age. So we need to be able to measure aging in individual cells. And then the fourth axiom is perhaps the most controversial, is that a biomarker of aging should apply to all mammalian species. And I say that because in my view, all mammalian species age. For example, we have managed to build epigenetic clocks for naked mole rats and pretty all mammalian species. Now, a lot of this talk is about axiom number 4, build biomarkers that work in all mammalian species. Now, to do that, we had to develop a particular genomic platform that focuses on 36,000 locations on the DNA molecule that are highly conserved in all mammalian species. And using this mammalian array, we are now able to build what I call third-generation clocks. For me, a third-generation epigenetic clock applies to multiple species at the same time. Before I go there, I want to talk about what I mean by relative age. Imagine you have species of different lifespan. For example, rats have a maximum lifespan of 4 years. So then the relative age of a 2-year-old rat is 2 divided by 4, 0.5. Conversely, a 61-year-old human has a relative age of 61 divided by 122, again 0.5. So we form this ratio in order to align different species. And so why do we do it? Because it's the simplest thing and it works. Is it the best way of doing it? Absolutely not. And there's an opportunity for the community. Build different measures of relative age, better alignment of chronologic ages. In any event, a lot of the talk will be about this simple model of relative age. And here I show you in the bottom a multi-species clock where on the left-hand side, human samples are colored in green and the orange samples are from a rat. But you see perfect alignment. So it's a flavor that you can build prediction models that predict relative age based on cytosine methylation. But let's come to the most ambitious question, which is it possible to construct epigenetic clocks that are applicable to every mammalian species? Mammals are separated by over 200 million years of evolution. You have bats, you have whales, different ecology. Can you build one clock, one regression model, one set of cytosines, no hidden parameters, really one math formula to estimate age? And the answer is yes. We just published this article in the journal Nature Aging. On the right-hand side, you see a panel where we predict relative age on the x-axis based on methylation. On the y-axis, every plot I show you is unbiased in the sense that we use cross-validation to arrive at unbiased accuracies. Correlation 0.95. To me, this is somewhat of a miracle of cytosine methylation that you can even do that. And it shows that there's something deeply conserved in all these species. There's some sort of clock ticking, and it does turn out to relate strongly to development. If you carefully look, the x-axis starts at 0. So from 0, a relative age of 0, all the way to maximum age, there's one clock ticking. Here I show you how this universal pan-mammalian clock applies to different species. Upper left panel, humans, correlation is above 0.95, mice, correlation 0.95, dolphins, dogs, elephants, bats. Remarkable conservation, one formula. But now I want to come to a curious finding, which is the following, or it's a question. Is it easier to build an epigenetic clock in a mouse or in a long-lived species, like a human or a whale? And the answer is, there is no difference. So the universal clocks, or more generally epigenetic clocks, are as accurate in short-lived species as in long-lived species. And this plot visualizes that. So the dashed line in the middle shows you the maximum lifespan of these different species. Species are arranged from short-lived to long-lived. And then you see on the outside this jaggedy curve. This is the accuracy of these epigenetic clocks, and there's no pattern. So yes, there's something invariant. Epigenetic clocks work equally well, irrespective of maximum lifespan. This talk mentions several interventions. OSKM reprogramming of human fibroblasts completely reverses epigenetic age. This is well known, but also for our universal clocks. Here's a more recent study with Harold Ketcher and Akshay Sanghavi, where we evaluated whether young pig plasma rejuvenates human rat clocks, so it's a multi-species clock. And when you compare the red bar, this is a tissue from an old rat, to the orange bar, which is the age of a treated old rat, you see drastic rejuvenation. And similar results have been obtained, of course, with parabiosis. Vadim Gladyshev talked about it yesterday. And I want to mention Vadim Gladyshev's lab has done more than anyone else to characterize perturbations that affect epigenetic clocks. And here I list a couple. Caloric restriction is a gold standard anti-aging intervention. Growth hormone receptor knockout, these dwarf mice age more slowly. Parabiosis, I mentioned. Young blood rejuvenates many organs even after detachment. But then pro-aging interventions exist as well. High-fat diet and certain genetic disorders, for example, Down syndrome. So we've now entered a new era of preclinical testing. We can apply the same epigenetic clock, the same conserved biology to several species that allows us to de-risk interventions. In other words, if we have an intervention such as AKG and it reverses a multi-species clock, chances would be that it also applies to humans, as an example. I now want to briefly talk about the mammalian methylation data, 15,000 tissues. What would you do if you're a data analyst? You start out by clustering the species. And Armin Hagani has done that. And what do you find? Well, if the species cluster by taxonomic order. No surprise, perhaps, except it's a striking finding because we focused on ultra-conserved cytosines. These cytosines do not differ in terms of sequence variation. It's strictly based on methylation levels in ultra-conserved cytosines. And this tells us something about evolution. Evolution clearly has an effect on the methylome. But the next thing a data analyst would perhaps do is to cluster the cytosines. Let's say you cluster 20,000 highly conserved cytosines. What do you find? Well, you find, in this case, these distinct branches on a hierarchical cluster tree. And these branches are known as modules. Module equals group of cytosines. And modules are colored, typically. And why do we do that? It's a data reduction method. You go from 20,000 cytosines to 55 modules. You will end up seeing the forest for the trees. So when you have a module, you can summarize it by a representative. We call it the module eigengene. And think of it as an average across the cytosines in the module. And that module eigengene on the y-axis correlates quite strongly then with maximum lifespan. Here, each dot is a different species, and you see that we have modules that correlate at a level of 0.55 with maximum lifespan. Interesting modules. But there's something curious going on, because we have on the right-hand side lifespan modules, and on the left-hand side we have what we call modules that respond to health interventions in mice, and they are different. So, for example, here we evaluate caloric restriction, and whatever caloric restriction affects in the methylome, these are different modules, different cytosines from those that relate to maximum lifespan. And of course, these methylation-based results very much match those from Vadim Gladyshev, who presented yesterday his analysis of gene expression data and in essence found the same thing. One way to interpret it is to say if you starve yourself, you will not affect maximum lifespan. That's according to this methylation readout. Now you can ask, well, which cytosines relate to maximum lifespan? What is known about the upstream regulators? And there's a surprise. We find some of these so-called Yamanaka factors. So these are factors that can turn cells into stem cells. And so we find these pluripotency factors. So very interestingly, this idea of regenerating cells or reprogramming cells does relate to maximum lifespan. And we observe also overlap at the level of genes when we overlap genes that relate to maximum lifespan with those that relate to changes following interrupted reprogramming with OSKM. Okay, several people have talked about maximum lifespan, and I want to comment a bit on it. So we built also predictors, methylation-based predictors of maximum lifespan. So these are very different from epigenetic clocks. They simply predict max lifespan in a species. And we had 348 species. That's the best predictor we could come up with. Hopefully your team develops a better one. But correlation 0.89 on the log scale. And to tell you the truth, I have no application of this maximum lifespan predictor. I mean, as a mathematician, I love it, but I have no use, except for one. I applied it to look at sex differences in different species. And out of all of these hundreds of species, we found 18 species where there was a difference in the predicted maximum lifespan. 18 species. Humans are one of them. And what does that mean for you if you want to extend your maximum lifespan? Turn into a woman. That's what it means, okay? Now I want to talk about another topic, EWAS. Here, what is EWAS? Epigenome-wide association study. Here we correlate individual cytosines to maximum lifespan. You've seen similar plots perhaps in genetic studies, so-called Manhattan plots, and we find so-called HOX genes that play a role in development. So many developmental programs. But let me give you the main punchline. The red curve shows you the methylation landscape of the DNA. Think of the x-axis as the DNA, the red curve is the methylation landscape of a long-lived species. And what do you see? Well, you see large peaks and deep valleys. So long-lived species have beautiful hilly landscapes. By contrast, short-lived species have landscapes that are more flat, more uniform, more entropy. All right, now I want to come to the final section of my talk, which is what I mentioned earlier. Cytosine methylation really allows you to bring the power of math into the longevity field. And so here I describe a preprint, fundamental equations linking methylation to maximum lifespan. So I'll start with some interesting results. Here I show you different species and they are arranged by maximum lifespan. The top is bowhead whale, 211 years old. Bottom is a shrew, can live up to 3 years. And the bars are the correlations of methylation with chronologic age. And what do you see? You see nothing. There's no pattern. And that's interesting to me because it shows that this age correlation could be interpreted somewhat as an invariant. You know, it doesn't relate to max lifespan. That's insight number one. Now I want to come to another idea that has been voiced many, many times. It goes back to the 1910s. 100 years ago, rate of change, rate of living hypothesis. But more precisely, imagine you measure the velocity of methylation increase in the DNA across the lifespan of a species, the rate of change in methylation. Well, does that relate to maximum lifespan? The answer will turn out to be yes. So here I show you in the left panel, I show you on the log scale how the rate of change of methylation, is the y-axis, correlates with maximum lifespan. Each dot is a species. And you see this beautiful linear relationship. And on the right-hand side, I remove the logs. So this simply means the average rate of change equals 1 divided by max lifespan. Very simple inverse relationship. So that's equation number 2. But let's be more precise. When you calculate a velocity, you're making an assumption. The assumption is that that the car or whatever doesn't accelerate. You assume a linear relationship. And here I show you that methylation, which is on the y-axis, actually has a non-linear relationship with chronologic age. So the green regression line shows you the rate of change in young animals, in young pigs, and the blue line shows you the rate of change in old animals, in old pigs. I could have shown you many species, many tissues. We observe that over and over. So if you then do a formal analysis of what I call the young average rate of change of methylation, which is on the x-axis, versus the old average rate of change in methylation, if you plot it, first of all, interestingly, you see a linear relationship, but also you see what I mentioned, that in the young animals, rate of change very much exceeds that of older animals. And that's in essence the third equation, the fundamental equation of methylation. So then here I show you all the data that we have. The y-axis shows you methylation, the x-axis shows you relative age, and you see some sort of a log linear relation, but the people who are better at math than me may be able to find a nice equation to link that. Yes, so to summarize that, we have these fundamental equations in linking methylation to lifespan. First of all, methylation levels in select chromatin states, in select regions of the DNA, exhibit this log linear relation with age across the entire lifespan. Then the equation number 1 is that the rate of change of methylation is 1 divided by max lifespan. Equation number 2 is that the correlation between methylation and age has no relation to maximum lifespan. Equation number 3 is that the young animals' rate of change equals that, or is proportional to that in old animals. And I want to give you one caveat. I talked about average rate of change in methylation, but it's actually very important to focus on the right regions of the DNA. And I didn't want to traumatize the audience, but they are so-called bivalent regulatory regions. These are crucial during development and cell differentiation, and you really need to focus on those regions. You cannot just pick random cytosines. I've started a non-profit foundation called the Epigenetic Clock Foundation, CEO Bobby Brook. So Bobby distributes the mammalian array that I described. It's available to all of you without any limitations. And I started the Clock Foundation to facilitate rigorous testing of epigenetic clocks. This is about the topic of let's prevent false findings in the literature. So in conclusion, cytosine methylation allows one to build these pan-mammalian clocks. Cytosine methylation allows one to estimate maximum lifespan. We have clocks for all model systems. We and others use clocks in clinical and preclinical studies. You saw the nice talk from Brian Kennedy about a clinical trial. And above all, I invite you to delve into our mammalian methylation data to unravel the enigma of maximum lifespan and aging. And here are some acknowledgements, and I stop. Thank you so much.
Thank you, Dr. Horvath. We have time for one or two questions, so got hands up in the audience. This one over here came up first.
Hi, Steve. Thanks so much. Great talk. Can you offer an interpretation of the rate of change in young animals being higher than in old animals?
I think, yeah, good question. Why is that? I don't. Well, people would say cell division, proliferation. That would be a theory. I don't. Probably it has something to do with it, but it could alternatively have to do with hormone exposures, right? During development, several hormones are in high abundance. And it ties in a bit with these findings that in these dwarf mice where growth hormone receptors have been knocked out, you see slowed aging, So we keep seeing a connection to hormones. I'm still struggling with that, but and then you saw these findings about young plasma circulating factors affect epigenetic clocks, So yeah, they are circulating factors, but sorry, there's so many hints, but really to nail it down is quite difficult and may depend on the clock that you're using. Clocks differ in terms of their biology. Yeah. Mm-hmm. Yeah.
Great talk, thank you. I just had one question. You got a lot of taxa, like 26 different taxa, but what tissue did all these samples come from? And do these clocks, even though they're highly conserved, are they highly conserved within an organism, within a tissue?
I want to say we analyzed 70 different tissues, pretty much every tissue you can name, or at least I can name. I can name 5 or 6, brain, heart, blood, skin, ears, kidney, lung, anyways, skeletal muscle, adipose. It differs a bit. For some species like horses, we have 60 different body parts. Then for other rare animals that come from, let's say, Saudi Arabia, we only had an ear sample, So we're not balanced. What I can tell you is, from our studies, we find that in general, brain ages more slowly, at least in humans. Also the retina, so that also ages more slowly, but it does depend a bit on the species. Mm-hmm. Yeah.
Hi, thank you for your talk. A question. Your company, your research lab is known as being cutting edge, leading the way, Altos Labs. Tell me, do you have a division on the future of technology, for example, with AI and nanomedicine? Is there any division of your lab that is looking at how nanorobotics on a molecular scale could be used for identifying methylation and epigenetics, et cetera, because that is the cutting edge of longevity? Thank you.
Yeah, so Altos Labs does not really work on longevity. It's a bit a misconception. Altos Lab really focuses on health span. So they want to boost the resilience of special cell types.
Then let me correct myself. For health span, let's remove longevity from it, or anti-aging from it. For health span, nanomedicine is at the cutting edge of what is going to be around the corner. And in looking at mathematics, looking at methylation, looking at all the research that's been done for eons with mice, eons, literally. What is at the cutting edge of Altos Labs in looking at not just AI, but nanomedicine on a molecular scale that could work more directly with robotics and AI-driven in identifying some of these biomarkers in methylation, et cetera.
Yeah. I really like your suggestions. I love it. Anyway, when it comes to company focus, we are an organization of 27 principal investigators. We have very different approaches. They range from imaging to genomics to everything else, mouse studies, so very broad. Mm-hmm. Yeah, thanks.
Steve, I have one question
Yeah.
about the methylation changes that happen as organisms age. Are those changes conserved, or is it random? So if you look at the methylation clock at different time points, are those changes happening randomly, or do you see a certain conservation of where those changes happen?
Yeah, it's really both. So it turns out the human DNA contains 28 million cytosines. Most of them are not conserved at all, and not even conserved across tissue type, meaning what changes in blood is very different from what changes in human liver. What machine learning taught us, though, is that there are certain select cytosines, select regions that where you do observe ultra-conservation, and these are linked to developmental processes, more specifically polycomb complex repression, and one needs to hone in on the right locations. Overall, bad conservation. Mm-hmm.
Thank you very much.
Yeah, thank you.